In amphibians, retinal ganglion cells, or RGCs, project their axons from the retina to the tectum along the optic nerve. When damage occurs to the optic nerve, amphibian RGCs have the amazing ability to regenerate axons and reestablish connections between the retina and tectum. In this diagram, the circle on the left represents the amphibian retina, and the oval on the right represents the tectum. The RGCs of the retina are represented in red, as are the corresponding axons that connect to the tectum. In a classic experiment, researchers transected the optic nerve, disconnecting the axons in the tectum to the retina. Then they ablated the RGCs in the dorsal half of the retina and only allowed the ventral half of RGCs to regenerate axons. These axons grew to innervate the medial half of the tectum. Conversely, when researchers ablated the ventral half and allowed only the dorsal half to regenerate their axons, these axons innervated the lateral half of the tectum. Likewise, posterior RGCs innervate the anterior tectum, whereas anterior RGCs innervate the posterior tectum. This experiment demonstrates that there is a point-to-point connection between the retina and the tectum, and it is through these ordered connections that a two-dimensional image in the retina can be reconstructed in the tectum. It is believed that during development, these point-to-point connections are directed by molecular gradients. An axon repellent, called ephrin A5, is expressed in the tectum with lower concentrations in the anterior region and higher concentrations in the posterior region, here represented in orange. In the retina, a receptor for ephrin A5, called FA3, is expressed with lower concentrations in the nasal region and higher concentrations in the temporal region, as represented in blue. Because temporal RGCs express a higher amount of FA3, they are more sensitive to the repellent and consequently project to the anterior region of the tectum, which contains a lower concentration of ephrin A5. Since nasal RGCs express a lower amount of FA3, they are less sensitive to ephrin A5 and can project to the posterior tectum. This proposed model was tested in a knockout experiment in mice, utilizing mutants of ephrin A5, as well as a second repellent molecule called ephrin A2. Both ephrin A5 and ephrin A2 exhibit a posterior high, anterior low gradient in the superior colliculus, the mammalian equivalent of the tectum. In wild-type mice, a focal injection of an axon tracer into the temporal RGCs showed that these axons terminate at the most anterior superior colliculus, as predicted by the model. In mutants with ephrin A2 knocked out, temporal axons shift their targeting more posteriorly. The results were similar when only ephrin A5 was knocked out. However, in mutants with both repellents knocked out, temporal axons did not exhibit any specificity along the anterior-posterior axis. These double knockout experiments demonstrated that ephrin A2 and ephrin A5 indeed play essential roles in vivo to instruct RGC axon targeting along the anterior-posterior axis of the superior colliculus.